My name is Kristiana Smith, and I am a junior attending the Baltimore Polytechnic Institute. I have had the opportunity to engage in research in molecular biology through the Ingenuity Project's Junior Research Practicum. Now, I will be sharing with you my project, Optimizing the Live Cell Imaging of Lamina-Associated Domains. Our bodies contain many different cell types, and they all share the same genetic blueprint, which is our DNA. Despite this uniformity in DNA, they are very different in function and characteristics. The characteristics and functions of a cell are determined by the activation of specific sets of genes that are necessary to that cell's function. Understanding how these genes are selectively expressed to yield such unique cell types is going to provide a more thorough understanding of how a cell's fate or role is established. Interestingly, the three-dimensional organization of the genome is thought to play a role in the activation or repression of specific genes, and understanding this process will contribute to a broader understanding of the relationship between epigenetics and cell differentiation. In a cell that is not dividing, we see normal organization. Normal organization consists of our chromatin being segregated into A and B compartments. Our A compartments contain our frequently transcribed DNA, or the genes that our cells use actively. B compartments, however, contain repressed portions of the genome and are also known as lamina-associated domains because they reside at the nuclear periphery, or nuclear lamina. The genes and LADs vary between cell type, and the variation is thought to guide cell fate and function. This figure shows general chromatin organization throughout the cell cycle. Our non-LADs are shown in blue, while our LADs are shown in magenta. Our non-LADs refer to the A compartment. Note how the A compartment tends to be directed towards the center of the nucleus. Conversely, LADs are compacted tightly against the lamins that are shown in green. This is what we consider normal organization. As the cell cycle progresses, this normal organization must be dismantled because mitotic chromosomes must form to carry out cell division. As cell division comes to a completion, normal organization is reestablished. It is unclear as to how this organization of A and B compartments is reestablished following cell division. Research in this field is primarily motivated by the intent to understand how genes are segregated into active and inactive regions. Currently, it is unclear how structures such as LADs are established after cell division, and an efficient method for visualizing LADs would provide insights on dynamics of their formation and placement. Additionally, the proteins involved in this process of establishing genome organization are largely unknown and are an area of interest. In an effort to identify LAD organizing factors, the Ready Lab used biotinylation identification and mass spectrometry to generate a list of proteins that may participate in LAD organization. Biotinylation identification, or BioID, is a method that detects protein-to-protein interaction. Members of the Ready Lab designed a protein, then as it was recruited to the nuclear periphery, proteins that interacted with it were reported. 2 proteins of interest were selected from this list due to time constraints and were visualized using fluorescent microscopy with the goal of assessing image quality. One protein is emrin and is a nuclear envelope protein that is anticipated to localize at the nuclear periphery. G9A is a methyltransferase expected to localize at repressed regions of the genome. G9A can transfer a methyl group or a chemical marker to different proteins in chromatin. A methyl group is a mark commonly associated with gene repression. This is why we expect that it would have a strong localization with LADs. Through indirect immunofluorescence and microscopy, we view the localization of several components of the nuclear landscape. We used lamin B1 to identify the nuclear lamina. We identify our LADs with the system, the M6A tracer system. We viewed our proteins of interest with fluorescent antibodies. We used HERXT, which is a dye that can help identify double-stranded DNA or DNA outside of LADs. and we then merged the images so that we can observe any overlaps in these components. MRIN has a clear and defined localization at the nuclear envelope as expected. However, G9A appeared to be nucleoplasmic and not specific to repressed portions of the genome or LADs at all, which was interesting to note. However, it's important to look at our LADs as we are gauging the quality of the M6A tracer system. These images showed that the M6A tracer system could be improved in order to enhance clarity of these images and future images alike. As shown by the images in the previous slide, our results indicated that there is a need for improvement in our current method used to label LADs, which is the M6A tracer system. The system consists of two constructs. The first is a fusion of the proteins DNA adenine methyltransferase and lamin B1. When this fusion protein is recruited to the nuclear lamina, it is able to methylate the adenine molecules in LADs. The second part of the system is another protein, M6A, which is conjugated to the M-cherry fluorophore, that is able to bind to the methylated adenine molecules and cause our LADs to be fluorescently labeled. It is now believed that through the addition of another construct called the halo tag and a fluorescent ligand replacing the M-cherry fluorophore, this will increase the stability of the system and enhance image quality. This figure illustrates the mechanics behind the M6A tracer system. Along with the fusion proteins, the system is coupled with several sequences such as a destabilization domain and the sequence known as the CDT-DAM lamin B1. These sequences, when coupled with the molecule shield, cause the M6A tracer system to have an increase in stability. As shown to the far right, without shield, the system is extremely unstable, while with the addition of shield, the system is exclusive to LADs. The addition of the halo tag will increase instability while negating the necessity for the shield molecule. In order to introduce the halo tag to the original M6A tracer system, a new sequence must be constructed. First, the original plasmid coding for the M6A tracer system will be cut with restriction enzymes BSTBI and BSRGI. This will allow for the M6A sequence itself to be isolated from the entire plasmid. Then, this sequence for the M6A portion of the construct will be introduced to the plasmid, coding for the halo tag itself. The halo tag sequence, or the PHTN-halo vector, was digested with a single restriction enzyme, EcoR5. It was then ran on a 1% agarose gel along with the uncut samples. Cutting with a single restriction enzyme allows for the circular plasmid to become linear and create space for the insertion of the M6A sequence. As we look at the gel above, we see that PHTN uncut 1 and 2 both ran a bit further than the sample that was cut with EcoR5. Circular DNA runs further on an agarose gel than linear DNA, so this indicates that the digestion was a success. Next, M-cherry M6A was digested with several enzymes. It was first digested with BSRGI and BSTBI. However, it was also digested with BSTGI and NHEI as an alternative restriction site. It was also ran on a gel to verify its success. Looking to the far right of the gel on the bottom, we see that 2 fragments were created by this digestion, so it indicates that M6A was successfully extracted through enzyme digestion. The resultant plasmid combining the M6A sequence and the halo tag sequence was digested with restriction enzymes KPN1 and BAMH1. This was done so that we could verify that the M6A and halo sequences were ligated together correctly and that the M6A sequence was inserted in the correct direction. However, when we look at this gel, we see many errors. Firstly, several of the columns are smeared. Secondly, in lanes 2 and 4, there are fragments visible, but it's unclear what they are. And lastly, the column with the PHTN EcoR5 cut sample has an additional fragment that should not be there. It's unclear where the mistake was made here, so this experiment will have to be repeated in order to determine which step in this process or in the cloning process had an error. In order to make steps to completing the cloning process, we must generate samples of all of the plasmids with a higher concentration of DNA, then re-verify that it is the correct sequence. We will redo the last experiment in order to determine whether mistakes were made and in which step they were made. The final M6A haloplasmid will then have to be inserted into a viral vector. It will have to be used to transfect in human embryonic kidney cells and produce a virus. This virus will then be used to transduce mouse embryonic fibroblasts, and this will incorporate the M6A halo sequence and construct into the new cell's DNA. In the future, we would like to do tests in mouse embryonic fibroblasts with the M6A halo construct, and this will help assess whether there are improvements in the labeling and visualization of LADs. We would also like to create similar constructs that label telomeric and centromeric repeats, so that we may investigate whether there are any interactions between telomeres, centromeres, and LADs. Lastly, the interactions between laminin proteins and the different types of heterochromatin will be explored in order to further investigate the dynamics of genome organization and the role that LADs play in genome scaffolding. I would like to acknowledge and express my gratitude to Dr. Karen Reddy and Victoria Hoskins, my mentors, and to my research instructor, Dr. Nicole Rosen, and to everyone at the Ingenuity Project. Thank you for your support and guidance.